and I'm going to present some experimental data combining behavioral measures, neuroimaging, and computational modeling to try to gather further understandings about the cortex basal ganglia loops and their role in cognitive and motor function. And how did you? Yeah, okay, now it works. All right, so subthalamic deep brain stimulation, we heard about that before, and we heard about the subthalamic nucleus and the basal ganglia anatomy in general, which is a great introduction to my talk, because then I believe that you can really understand where in the subcortex we are. And it is known that this therapy is a very effective treatment alternative for patients with Parkinson's disease that do not have satisfactory clinical symptom alleviations through medication alone. And it is a standard treatment for many patients, and stimulation in these patients is chronically applied through an implantable device that is either in the abdominal wall or the thorax of the patient and connected to the intracranial leads. And because it's a reversible treatment, you can switch off the stimulation, you switch it on. You can also record, as we heard before, from the electrodes. It is a unique research platform to study the physiology and pathophysiology of Parkinson's disease and basal ganglia. And what we have learned from the 30 years of research in lesion and DBS for Parkinson's disease is that one of the mechanisms of DBS may be some sort of net inhibition in the basal ganglia thalamocortical loop and in the motor network. But as we know now through lots of different studies and really defined by Adam Aaron, who is the organizer, the subthalamic nucleus is also part of a cognitive triangle and may be involved in a direct or a fast way to global inhibition directed by frontal cortex. And this has been replicated many times in perceptual, in different cognitive decision-making tasks, that if you have higher conflict in a task or a task is more difficult, then STN DBS compared to stimulation turned on versus turned off leads to faster responses only in the difficult condition. And you can see that easily on the right side, where the strongest DBS effect is on the left side, where the decision that the patients had to make was the most difficult one, whereas on the right side here, in this moving dots task, all dots were moving really coherently, and it was incoherently, and it was very easy to make a choice. And then DBS did not have a reaction time effect. So there is experimental evidence for the role of the subthalamic nucleus in response inhibition. And when we're talking about inhibition, a clinician may be talking about something different to what I've introduced on the cognitive side before. And we actually know that the subthalamic nucleus is part of two pathways of inhibition. So the subthalamic nucleus itself is mainly glutamatergic. It projects to the internal pallidum, which is the main output structure of the human basal ganglia. And the internal pallidum is chronically active, chronically inhibiting the thalamus. And the subthalamic nucleus, through glutamatergic projections, can increase this pallidothalamic GABAergic output, and increasing subthalamic glutamatergic output can therefore increase global inhibition signals of the cortex basal ganglia thalamus network, while DBS or lesioning the STN may decrease this this activity. And we have the cortico-subthalamic projection, which has been hypothesized to play a role in cognitive and inhibitory control. And we also have the indirect pathway, which is known to be modulated by dopamine. And in Parkinson's disease, we have a hypodopaminergic state, which leads to an increase of indirect pathway activity, and therefore to a global increase of net inhibition from the basal ganglia onto the thalamus. So my interest was to try and see whether we can segregate hyperdirect and indirect pathway DBS effects in patients. And therefore, we designed a behavioral task where we combined more difficult and an easier condition on the cognitive side, or at least on the motor preparation side, and combine that with motor output that we can measure. And on the left side, you can see here, patients were asked to steer a cursor on a tablet, steer a pen on a tablet to reach a target on the screen with the cursor. And in the left condition, it was the mapping of the pen to the cursor was unperturbed, while on the right side, what we call the controlled condition, this mapping of pen movement to cursor movement was inverted. And we included patients off DBS and switched the stimulation on in a randomized fashion. And we also included healthy controls. And here you can see on the right side first that there's a huge clinical improvement in these patients through the stimulation. UPDRS-3 is a measure of symptom severity in Parkinson's disease. And this cohort was 20 patients which had a great clinical effect. So we expected also a good behavioral effect in the study. And now we compared different measures of behavior. On the left side, first, you can see the reaction times, and blue is always off DBS, red is on DBS, and yellow is healthy controls. And we could replicate the cognitive effect that was shown before, that when there's a more difficult condition, DBS creates a stronger reaction time effect. As you can see in the easy condition, there's no significant difference between off and on DBS, while in the controlled or more difficult inverted pen to cursor mapping condition, there's a significant change in reaction time and reduction in reaction time. Regardless or independent of that, we could look at the actual kinematics of the movements that were done, and we could show that DBS leads to a profound increase in movement velocity. It almost doubles the speed that patients could move, and that was to the level of healthy controls, actually. But what is interesting, if you compare the two conditions that had a dissociation on the reaction time, there's no dissociation at all in Parkinson's disease through the condition, while healthy controls actually slow down their movements to adapt to the more difficult condition. And that may result or at least correlate with larger error or more erroneous movements. So basically, we have a dissociable cognitive effect or reaction time motor preparation effect from the actual movement kinematics. And these may be related to the different pathways. And the next thing that we did is looked whether, from an imaging perspective, there's evidence that cortico-subthalamic projections can explain the reaction times effect. So we localized the electrodes in these patients and mapped them to MRI space using the lead DBS toolbox, which was developed in our center by Andreas Horn. And we conducted fiber tracking in a normative connectome in MRI space and looked where the active contacts resided with relation to the SDN and with relation to fibers coming from sensory motor or cortical and premotor cortical areas, and created this patient connectome and here you can see all the fibers that were found to be connected to active contacts throughout the whole cohort. And indeed, we did find a correlation with reaction time effect with with the density of fibers that we are stimulating in each patient. So the more sensory motor to STN fibers were affected by the stimulation, or the closer the active contact was to these fibers, the stronger the reaction time effect was. And when we looked on the cortical side, we found that fibers coming from the SMA were the most predictive in terms of the reaction time effect. Importantly, there was no correlation for the clinical effect with this with fiber tracking, and we controlled for clinical symptom alleviation and also for movement kinematics that I showed before, and we did not find any association of that. So then we thought, well, how can we further evaluate the subcortical part? And we utilized a whole network firing rate model based on the presumed architecture that we heard before. And we trained this model to perform the task. And here is an example of a healthy subject performing the task in silico. There's the cue coming up soon here on the left side, on the right side, and then you can see cortex activation leading to the full pipeline of cortex basal ganglia computations, ultimately resulting in movement of the cursor from motor cortex activity. So we could look at the activity of the model, computational model, and we could perturb the model in ways that we believe the DBS could actually modulate behavior in humans. And we informed these models by the results of the fiber tracking, but also by clinical measures like the change in UPDS from DBS, and also the UPDS off and on scores. So we looked whether indirect versus hyperdirect pathway lesions to the model, because that is a simplified way of thinking about what DBS does, could predict our behavior in patients. And we found that indeed hyperdirect pathway lesion predicted reaction time effect in the task best, while indirect pathway lesions predicted the movement time effect best. And combining all measures, the combination of a block of hyperdirect and indirect pathway activity could best predict behavior and the whole performance of the task. So to conclude, we have shown evidence that subthalamic stimulation may affect different pathways at the same time, which is kind of an obvious statement, but we have shown or tried to dissect which effects these simultaneous modulations of different pathways can have on the basal ganglia and have on the capacity to move and to behave in PD patients. And we believe that cortical subthalamic hyperdirect pathway stimulation may lead to create decreased reaction time adaptation. That is an independent effect from the clinical improvement. And this can be useful at some point in the future, because we may be able to create computational basal ganglia prosthesis. And this is, I think, where far off DBS researchers are looking, or where DBS researchers are looking forward to try to do a stimulation paradigm that learns the demand of stimulation from a combination of computational basal ganglia models and artificial intelligence to cater the individual stimulation to the patient's needs and also to the physiological function of the basal ganglia that we're trying to modulate through DBS. So the first study I showed is published in Brain, and I just want to give you a heads up if you're interested in basal ganglia cerebellar connectivity. We also got a recent paper on that coming from with a similar paradigm. And I thank you for your attention. I would like to thank our collaborators and Anna, who has performed the behavioral tests. Andy has shown me and helped a lot with connectomic analysis, and of course Professor Kuhn, my PI and supervisor, and Adam Aaron for organizing this great symposium. Thank you.